Northern European. North European ancestry reflects long-term adaptation to cold continental and maritime climates with limited winter sunlight. Low melanin pigmentation supported vitamin D synthesis in low UV regions, while metabolic traits favored efficient energy storage during long, resource-scarce winters. Facial features often include narrower nasal passages and a more elongated mid-face, helping warm and humidify cold, dry air before it enters the lungs. High dairy reliance over millennia led to strong selection for lactase persistence, allowing adults to digest milk as a stable calorie source. Immune-related diversity expanded through repeated migration waves and cultural mixing across northern forests, river basins, and open plains. Coastal populations developed traits tied to fat metabolism and cold water exposure, while inland groups adapted to grain-based diets and seasonal temperature extremes. Overall, North European ancestry represents a broad, climate-driven genetic profile shaped by low sunlight, cold seasons, and long settlement continuity. Sub-Saharan African. Sub-Saharan African ancestry contains the greatest genetic diversity, creating a wide range of adaptations tied to heat tolerance, pathogen exposure, and physical performance. Because humans have lived in this region the longest, many inherited patterns are shaped by hot climates and high disease pressure, appearing as population trends rather than fixed traits in every individual. Heat management is a core feature. Variants influencing sweat efficiency, strong skin barrier function, and higher melanin levels help protect against intense sunlight and maintain cellular stability under strong UV radiation. Immune adaptations reflect long exposure to parasites and rapidly evolving viruses. A broad array of HLA variants improves pathogen recognition, while certain blood cell traits historically reduced malaria severity by disrupting the parasite's life cycle. Musculoskeletal and metabolic tendencies also show long-term mobility demands. Some variants support greater baseline power or speed, while efficient glucose use aligns with environments where high physical activity was a routine. North African and Middle Eastern. North African and Middle Eastern ancestry reflects adaptation to dry climates, strong sunlight, variable food access, and dense trade-linked populations. These pressures shape traits that improve heat tolerance, water conservation, and immune flexibility. Hydration control is a key feature. Variants affecting sweat rate, kidney water reabsorption, and salt balance help reduce fluid loss and stabilize blood pressure in desert conditions. Metabolic conservation also played a role. In regions where food availability shifted with seasons or trade, some variants supported efficient fat storage and slower energy use, helping people maintain strength during long periods of scarcity. Immune traits show the influence of historic trade routes. Exposure to pathogens from multiple regions selected for immune variants capable of recognizing a wide range of bacteria and viruses. Overall, this ancestry reflects heat resilience, resource efficiency, and broad-spectrum immunity. South Asian. South Asian ancestry reflects adaptation to monsoon climates, dense populations, high pathogen exposure, and long agricultural histories. These pressures shape traits linked to heat-humidity tolerance, digestive efficiency, immune response, and nutrient regulation. One major pattern is the ability to function in hot, humid environments where sweating is less effective. Variants affecting sweat composition, skin barrier stability, and moisture retention help maintain body temperature despite reduced evaporation during monsoon seasons. Immune traits show long-term exposure to waterborne and parasitic infections. Certain HLA and receptor variants support faster recognition of gastrointestinal pathogens and vector-borne diseases that spike with seasonal rainfall. These balanced immune responses are crucial in regions where infections reappear annually. Dietary adaptations developed alongside rice and wheat-based farming. Many populations in this region carry variants that improve starch digestion, glucose transport, and insulin efficiency, fitting diets built around carbohydrate-heavy staples. Nutrient regulation also reflects local environmental pressures. Variants influencing iron absorption, folate pathways, and UV-related metabolism help maintain stable nutrient levels despite soil variability, intense sunlight, and historically vegetarian lean diets. East Asian. East Asian ancestry reflects adaptation to temperate to cold climates, long-term rice agriculture, dense populations, and recurring respiratory infections. These pressures shape traits involving skin resilience, alcohol metabolism, carbohydrate processing, and immune response to airborne pathogens. Variants in genes such as EDAR influence thicker hair, stronger skin barrier function, and better moisture retention, useful in colder, drier seasons common across northern and Central East Asia. Alcohol metabolism is another notable feature. Variants in ALDH2 and ADH1B speed the initial breakdown of alcohol but slow acetaldehyde clearance, causing the well-known flushing response. Although uncomfortable, this effect historically discouraged heavy alcohol intake and lowered pathogen exposure from early fermented beverages. Long-term rice-based diets selected for efficient carbohydrate use. Variants regulating amylase production and glucose transport help maintain stable energy levels in populations dependent on high glycemic staple crops. Immune traits reflect life in dense settlements. Certain HLA types and receptor variants enhance early detection of respiratory viruses while keeping inflammation moderated, an important balance in regions where airborne infections circulated frequently. Southeast Asian ancestry reflects adaptation to tropical rainforests, constant humidity, and ecosystems rich in insects, plants, and parasites. These pressures shape traits tied to moisture regulation, parasite-focused immunity, flexible metabolism, and efficient detoxification of plant chemicals. High humidity is a major force here. Variants affecting sweat composition, skin barrier lipids, and microbial resistance help maintain cooling when evaporation is limited and reduce the risk of fungal or bacterial skin infections. Immune patterns show long exposure to parasites and vector-borne diseases. Many populations carry HLA and cytokine variants that create a balanced response, strong enough to control infections but moderated to avoid chronic inflammation in environments with year-round pathogen pressure. Dietary adaptations developed from mixed land and water food sources. Rice, taro, and yams provided steady carbohydrates, while fish and shellfish supplied essential fats. Variants influencing amylase activity, glucose transport, and liver detoxification pathways, including cytochrome P450 enzymes, support this versatile metabolic profile. Mediterranean European ancestry reflects adaptation to warm, sun-intense climates, diverse coastal diets, and thousands of years of dense agriculture and trade. Moderate melanin pigmentation balances UV protection with strong vitamin D synthesis, while skin barrier and sweat-related traits support hydration and heat tolerance during long, dry summers. Diet shaped many metabolic tendencies. Variants that efficiently process monounsaturated and omega-3 fats align with olive and seafood-rich nutrition, while carbohydrate-related adaptations reflect millennia of wheat, barley, and later rice cultivation. Because Mediterranean societies connected Africa, the Middle East, and Europe, immune systems were shaped by constant pathogen exchange, favoring broad HLA diversity and flexible inflammatory responses. Additional traits linked to cardiovascular efficiency, salt balance, and muscle endurance developed in populations reliant on coastal labor, sailing, and year-round physical activity. Together, these adaptations form a profile built on bright sunlight, maritime ecology, and the deeply interconnected history of the Mediterranean basin. Native American ancestry reflects one of the oldest and most isolated human expansions outside Africa. Early populations crossed the Bering Land Bridge during the last ice age, moving through Arctic tundra, Pacific coasts, and deep continental interiors. This long migration shaped traits centered on cold endurance, metabolic efficiency, and immune adaptation to low pathogen environments. Many communities developed variants supporting fat metabolism, glucose regulation, and higher brown fat activation, useful for survival in sub-zero climates and long-distance travel. Pigmentation remained generally medium to dark because UV exposure varied across latitudes, from equatorial Andes to northern Canada. Facial structure trends, broad cheekbones, robust midface, are linked to cold air warming and developmental adaptations from ancestral Siberian populations. Genetic diversity later expanded as groups adapted to rainforest humidity, Andean altitude, or Great Plains drought. Altitude-adapted communities, especially in the Andes, show strong selection for oxygen transport and elevated hemoglobin efficiency. Meanwhile, Amazonian groups display immune traits shaped by dense biodiversity and high pathogen environments. Across both North and South America, Native American ancestry represents a spectrum of environmental responses layered on an ancient Siberian foundation, producing a lineage shaped by isolation, climate extremes, and region-specific ecological pressures. Pacific Islander and broader Oceanian. Pacific Islander and broader Oceanian ancestry reflects adaptation to isolated islands, long-distance ocean travel, and diets centered on starchy root crops and marine foods. These environments shape traits tied to muscle performance, water-salt balance, carbohydrate metabolism, and immune patterns influenced by small, closed populations. Many groups show tendencies towards strong, power-oriented muscle function, supported by variants affecting fast-twitch fibers and mitochondrial efficiency, useful for paddling, hauling, and navigating open seas. Water conservation was also crucial. Traits enhancing kidney reabsorption and electrolyte stability helped maintain hydration in humid, high-heat environments with limited fresh water. Diet-based adaptation includes efficient processing of taro, yam, breadfruit, and seafood, with higher amylase activity aiding starch breakdown and omega-3 metabolism supporting energy stability. Immune diversity trends remain narrower due to geographic isolation, where fewer but persistent local pathogens shape selection pressures. Overall, this ancestry represents a blend of maritime endurance, island ecology, and nutritional specialization. Don't just watch the world, subscribe and understand it.